So the idea of this COVID diagnosis context is that the students have to diagnose whether or not 8 individual patients have got COVID and which particular strain they might have. And they're able to identify the strain using different primer pairs. So, for example, the wild type strain can be amplified by the green forward and reverse primers, and indeed the blue and the yellow ones as well. But this phi strain here wouldn't be amplified by the yellow primer because there's been an insertion there. And in order to identify the phi strain, we would need a new special primer, this magenta primer here. So basically different strains and different primer pairs are going to give amplicons or not, and those amplicons may vary in size depending on the pairs. So if you were to use, say, the green forward with the green reverse, you'd get one particular length. If you use the green forward with the yellow reverse, you'd get something a bit longer. Now, this becomes relevant because although Tara, who created the idea of this with me, has made 3 different variants that we tell the students about, there is in fact a tantalizing space here for another variant, and that's a little curveball that the students are thrown as they perform the analyses. Now, the actual sequences that go to create those that sort of graphic are held in more detail in the actual sequence data, and the students have access to that. So when they start up the task, they're going to create all the tubes that they need, and that of course are the 8 patient samples. And as you might expect, these patient samples are randomized in their composition between every single run. So every student has the same strategic objective, but every student has a completely different outcome in terms of what each patient has been infected with. So there can't be any plagiarism at all. We have the different primers, the 4 forward primers, green, blue, yellow and magenta, and the 4 reverse primers as well. And then we've got some cDNAs of each of the different types of COVID, wild type Phi and Phi B2. And we also have some ordinary human cDNA, which could be useful for doing some controls. And then, of course, we've got the real-time buffer. So one way in which a student might go about this is that they might create a plate with, say, 9 columns and 1 row, and they might pipette into each of these wells the forward green and the reverse green primers. And a standard setup is 5 microliters of each of the primers. Now, I'm going to be a bit fast about this because I know that you don't particularly want to see all the pipetting actions. But really, of course, students should be changing the tips. They should be not talking as they're doing this because it can get very confusing. It's not too bad when there's just a couple of wells that have been filled up, because you can see which ones have got some liquid in and which ones haven't, but it all becomes very fraught as soon as you've got the wells with something in. So I'll put the forward primers into each. I'll be good and generate some reverse primers for the green into each one. I guess we could have helped people by making these solutions green, but I think they can cope with the fact that they're only green by name. Now, as I've been talking, of course, I've actually forgotten whether or not I've actually pipetted things into some of these tubes, so I could have got this wrong, but I think I've got all the primers in there. I'm going to now put 10 microliters of buffer into each tube. So let's put 10 microliters of the master mix buffer. That is obviously contains all the triphosphonucleotides and the polymerase enzymes that are required to do the reaction. Again, I'm not paying particular attention to what I've put in. I think I've got everything in each well, but we'll soon find out if I have made a mistake. And finally, I'm going to put each of these patient samples, 5 microliters of these, into each of the first 8 wells. So A goes in here. Now I should do a new tip now, but because otherwise I might get some contamination. But I'm not going to, just for the sake of speeding through this, because all we really want here is an idea of how this works. And then the very final one, number 9, I could leave blank as a sort of control. I could put human cDNA in. I'm going to put wild type in there because I want to just check that these green primers do work with the wild type. But I could have made a couple of extra wells, one for human as a sort of negative control, one with nothing in as a real negative control, that sort of thing. Anyway, let's mix those, and then all we do for running the qPCR is we literally just press this button up here, place in, and that generates all of the amplification data. We copy that now and take it into Excel. Now, all we have to do there is create a new worksheet, and with this worksheet we can paste the data in, and for each of the tubes we've got values for how much amplicon there is in each cycle. So you can actually plot that out quite easily, but some of the real interesting information is right at the end. What is the final amount of amplicon? So millions of amplicons in these samples, not so much in tubes 1 and 6, and demonstrable lengths of amplicon in these. So it's amplicons that are 115 long or 109 long in these. And then TMs, melting temperatures, whoops, let's go back. Just one species here, 2 species here, 3 species in this one. So that starts to give us a clue as to what might be in those samples. Now, as I mentioned, the students do have access to this data here, which shows how each primer was designed and therefore what length each amplicon should be. I'm not sure whether Tara gives the students this cheat sheet, but this is what she and I would refer to when we were checking that everything was working okay. And basically you can see there that what we've managed to produce are amplicons of 115 and 109, and 115 is the classic for green forward and reverse for the wild type, the alpha strain. And 109 is what you would get with the green primers if you had Phi or Phi A2. So we immediately can start piecing together what must be there, what these TMs must represent. And so as we go through the puzzle, as we go through other iterations, try other primer pairs, then you can see that we start to build up a really good picture of what each of these samples must contain. Remember, we've got other primer pairs, other cDNAs that we can use. So it becomes a really valuable student assessment. It's completely non-plagiarisable. There's lots of reasoning to do, but they also have to get the design piece done properly. They have to get the execution piece done. And it's only really if they've got data that looks robust that then they can then go ahead and start trying to piece together which COVID species is in each of these tubes. So extremely valuable, and we ran that assessment with 100 or so students, quite easy to mark, and really good discrimination between students who are willing to put the work in and do things properly and those that just wanted to adopt a perfunctory approach.